Welcome to MOOC course on Introduction to Proteogenomics. After understanding the two approaches for pathway enrichment and basic differences between both the approaches, we will now listen Dr. Carsten Krug, who will explain the use of GSEA at the pathway level analysis for different PTMs by taking an example of phosphodata. Dr. Krug will talk about the way one could analyze the available data at gene level to take it to the PTM level. He will also talk about the recent work, which is an initiative to make a PTM site curated database at Broad Institute. This involves a scoring of each P site by mapping it against the database. So, let us now welcome Dr. Carsten to talk more about how GSEA can be used to map PTM pathways for the analysis.
So, now I am going to talk about phosphodata, but this is actually true for all kinds of PTMs. So, there we measure different phosphorylation sites on proteins, right? And it might happen that we have multiple phosphosites on the same protein. It might happen that we have different isoforms of the same gene or protein, you know, and we measure different phosphosites on that, and so on and so forth. But in order to do pathway analysis, all of these pathway databases are gene-centric. As I said, pathway is a list of genes. So right now, all of the database that we have are curated at a gene level. So meaning if we want to do any pathway level analysis of our phospho data, we first have to collapse all of these measurements of phosphosites into gene level. So we're basically throwing away a lot of information. But this is something we have to deal with right now because there are no databases that are curated at PTM site level or at protein or isoform level. So you would do that by, for example, taking the average per gene or median, or looking across the most variable site, you know, across your samples. So variance means information, so that's why you would pick that one. So this is one example where we have employed this approach. This is again from the breast cancer study published in Nature 2 years back, where we started with roughly 6,000 phosphoproteins and performed the same type of analysis I just said. So we used single sample GSEA to map these phosphoproteins, which we of course first collapsed to genes using a median ratio and so on and so forth. We projected those into the space of pathways, 900 pathways. And then we performed consensus clustering on this data matrix. And interestingly, we saw like a clustering, like a unique cluster which we only saw in pathway space, and we would not have seen that if we would look at phosphosite or phosphoprotein level. So that's one example where these kind of analysis, where you just project your data onto a higher level of annotation and perform some analysis, can give you new insights that you would probably have missed if you would not have done that.
So please can you repeat, you mean to say according to the phosphorylation site?
Can you please use the mic that everybody can understand you?
So just wanted to understand, you are saying according to each gene and or according to our biological function, it will arrange the phosphosites.
No, here we are not looking at phosphosites anymore. So
no, you are clustering that phosphosites in each signaling pathway. Isn't it?
No, we calculate for each. I mean, now we're not looking at phosphosites, but each row is a pathway. And the data is actually the enrichment score I was describing a couple of slides back.
Yeah.
So we already combined all phosphosites to phosphoproteins to genes, and then we perform our signature projection method. So meaning in each sample, so these are the 77 breast cancer samples that we are looking at here, and in each sample we have a score, an enrichment score for this particular pathway, right? So red means this pathway is more active, Blue means this pathway is less active in this particular sample. And then we can do, in this case, we've done an unsupervised cluster analysis on that. So a very convenient way to, combine multiple measurements of phosphosites that map to the same gene to a gene-centric level is actually Morpheus, which is a very versatile matrix visualization tool. We're going to use it again in the hands-on. It's very powerful, but we are only going to use it for this particular purpose here. So just like to repeat what I've just said, so we start with a data matrix, which can be either proteins, phosphosites, or genes, or transcripts, whatsoever. But the first step that we always have to do is we have to roll up our expression values to the level of genes. Because all of these databases that we are using for our pathway analysis are gene-centric. And after we have done that, then we can continue with our pathway analysis, employing GSCA, single-sample GSCA, David, or there's so many different tools and approaches out there. It's, a matter of taste, what you like more, but we highly recommend and we highly prefer doing some sort of GSCA, genes enrichment type of analysis, because you don't have to throw away, you don't have to filter your list beforehand. So as I just told you, all of these databases that are available now are curated at the gene-centric level. And I just want to introduce you to like a project that we are doing at the Broad together with many other collaborators, where we actually try to come up with a pathway database that is curated at the site level, at the phosphosite level. So this involves many people and many resources. Okay, let's stay here for a moment. So we call that the PTM signatures database, which in theory, that's actually the goal, to be able to score each and every single phosphorylation site directly against the database. And that's a large curation effort, as you can imagine. It's very difficult to curate pathways at the gene level, but if you want to even go deeper and want to curate at every single phosphorylation site, what does it do in this pathway? Does it go up or down, and is it involved at all? So it's a lot of curation effort.
According to this is a human data only.
And most of the signatures are human. We started to do that for mouse and rat too, but it's mostly human. So we teamed up with other database curators from Phosphosite Plus, from NetPath, also WikiPathways, as I mentioned earlier. And with the help of all of these people involved here, we started to curate this database. Another very important aspect when you want to do something like this is, how do I represent a PDM site robustly? Which might sound trivial, but it's actually like a big problem. Like gene symbols have been standardized in a way, right? There are these Hugo gene symbols which try to harmonize and standardize human gene symbols. If you look into protein databases, the same protein might have a different accession number in one database, so this is now a UniProt database. If you look up the same protein in RefSeq, it will have a completely different, unrelated ID, so it's very difficult to cross-reference those, right? And this is even more, even more severe problem if you look at PDM sites. So how do you robustly represent a PDM site? So there's different ways how we try to approach this problem. So one is UniProt centric. So UniProt is a highly curated protein database. So we picked UniProt and we represent the site as UniProt ID, the modified residue, and the PDM type. And in this database, we also have information whether it goes up or down in a specific pathway or perturbation. So another way to represent phosphosite are flanking sequences. So this is what we just looked at in the morning, right? So we look at like plus minus 6 or 7 amino acids or whatsoever, around the phosphorylation site. This is a pretty unique identify already if you compare that in the human proteome. And also phosphocyte they are actually trying to come up with an unambiguous way to group sites across or within protein families. Just keep in mind, this residue number might change if you look at isoform A or isoform B, right? It might be the same site, but the residue number might be completely different. So the site group ID tries to harmonize those kind of events. Okay, so I'll quickly go through here. So right now we have like pathways, we have kinase substrate signatures, so we talked about, and we have a lot of perturbations by growth factors, also small molecules in this database. And so most of them, like these pathways, have been completely manually curated by curators from NetPath, WikiPathways, and we also extracted signatures semi-automatically and fully automatically. So we try to come up with a standardized way how to extract automatically derive these kind of signatures from known literature. Let me quickly go through these slides here. So, and actually, in order to extract these signatures, we came up with the method to extract consensus signatures. So let's say you have one perturbagen or one pathway that has been studied by different studies, by different labs. And there's different papers that might report, this site goes up on this perturbation. Another study reports the same site, but it goes down. So this kind of inconsistency you find all over the place, right? This might be due to, the labs, they use different cell types, experimental conditions, different protocols, or whatsoever. So what we try to do, we try to come up with a consensus between at least two independently published papers, in order to include these signatures in our database. And we use this very similar approach compared to GSA. We actually extended that scoring scheme in order to look for these signatures. So we tested that against a very well studied data set of EGF stimulated HeLa cells, has been published a couple of years back now in 2014. It's a very good systems biology data set to test your computational tools. So we picked that one because the authors used nocodazole to mitotically arrest HeLa cells, and also EGF to stimulate phosphorylation in general signaling. And both of these signatures are in our database. So this was our benchmark data sets. If we can pick up these signatures, I think we are on a good track that what we are doing is the right way. And what you're looking here is the heat map of enrichment scores. So each row here is an enrichment score of a signature, and here are the different experimental conditions. Red is DMSO, blue is EGF, green is nocodazole, measured in different number of replicates, right? So here's 4 replicates, here it's 6 replicates. And luckily we see that nocodazole here in green, the highest enrichment actually we observe in the nocodazole treated samples. So that's a good thing, and the same is true for EGF, right? And also in the control sample, we don't see any consistent enrichment. So this was our kind of global approach to prove that we are on a good track. And then we specifically focused on the clustering metric. So how well do these enrichment score cluster our data? And we compared it to a gene-centric approach. So here on the left, we look at this is how clean our clustering is if we do a site-centric approach, and this is how clean our clustering is if we first project it to genes, then do the same type of analysis. And we see that in a site-centric type of analysis, we see very clean clusters. So the higher these bars are, the better representative is my clustering. So and also what we can see in the gene-centric space is that samples from the control and the DMSO are clustering together, which is not what we would expect. And also if you just look at these EGF and Nakoda signatures alone, so and compare the signature scores across these different treatments and compare site-centric and gene-centric, we definitely see that we see a higher enrichment compared to gene-centric approach in the site-centric approach for EGF and also for Nakoda. So meaning we can pick up, or we have a better signal of the biological pathways if we do it site-centric compared to gene-centric. And I think now I'm going to wrap up here. So these tools are available on GitHub and GenePattern. There's many other tools that do similar kind of things like FoxTracks that specifically look at kinase substrate interactions. There's no other database that can do pathway or perturbation analysis. But there's other databases that do kind of substrate analysis, as we've talked about that this morning. And tomorrow, I think you're going to hear about WebGestalt and so on and so forth. So, and with this, I want to end my talk. Again, here I put some references, and I'm open for 1 or 2 questions before Bing is going to talk about. Any questions?
In PTM's SIG database, how you are taking care of isoforms in Uniprot number that you have shown here?
Yes.
So if we have, suppose for breaker, if we have 12 isoforms, so there will be the hyphen 1, 2. So can we give our data in that format?
So we recommend to use the sequence windows, which I know that you don't have in your database, in your data set. But the sequence, if you just represent a phosphosite by its sequence window, by the flanking sequence, it's much more robust identifier.
In this tool also we have to
Yes.
In this tool also we have to give the sequence window.
You don't have to, but you're on the safer side if you do so.
So today, in conclusions, you learnt about GSEA and how it can be used for pathway analysis for all the different kinds of PTMs. We also learned that all the pathways are gene-centric, all the databases are gene-curated, which may dilute your efforts. Hence, we need a PTM-curated database for proper analysis of PTM data studies. We also heard that why different IDs and different isoforms curation is important, but very challenging. We also heard about the 3 signature categories of PTM SIG database. For example, perturbational signatures, signatures of molecular signaling pathways, and signature of kinase substrate interactions. Curation of PTM sites using GSEA can be done manually by semi-automated or fully automated function. Based on this, they have also made PTM SEA or the modified version of GSEA to look at the signatures of PTMs. We also saw with an example that site-centric data grouping is more efficient and properly grouped as compared to the gene-centric. The next lecture is going to be hands-on exercise by Dr. Kirsten to show us how one can use GSEA for pathway enrichment. Thank you.